Welcome to the deep dive. Today, we're jumping into a really crucial moment in biology. For years after Mendel, these factors he talked about, the things carrying traits, they were completely abstract.
Just concepts.
Exactly. So our mission today is to trace how scientists went from those ideas to actually finding their physical location inside the cell. It's really the foundation of modern genetics.
That's right. We're talking about the chromosomal basis of Mendelism. It happened early 20th century, and it was really a fusion of two fields. You had cytology, the study of cells and genetics coming together.
Right.
People like T.H. Morgan, Edmund, Beecher, Wilson. Their work showed that how chromosomes behave during meiosis, well, it perfectly lines up with Mendel's laws.
Okay, let's start with what they could actually see. The chromosomes themselves. W. Waldeyer coined the name, didn't he?
He did, back in the 19th century. And the key thing is you only really see them as these distinct sort of dense structures when the cell is dividing. Otherwise, they're relaxed, more like diffuse strands called chromatin. But during division, they condense, and you can count them. Look at their shapes. And that number is important. We talk about ploidy.
Ploidy. Okay, so if I'm looking at, say, a human skin cell, what's the ploidy there?
That's a diploid cell, or $2. It means it has two full sets of chromosomes, one set from mom, one from dad.
46 total in humans.
46 total. The basic number nonetheless is the haploid number. You find that in gametes, eggs, and sperm. So 23 in humans.
And this number varies a lot between species.
Oh, wildly. You've got muntjac deer with just, I think, 3 pairs, and then some ferns with hundreds. But the basic idea of diploid body cells having 2 homologous copies, that's pretty standard.
Got it. Now, the structures that really provided that first tangible link between Mendel and the cell, those were the sex chromosomes, right?
Absolutely. Early cytologists like Wilson noticed differences between male and female cells.
What kind of differences?
Well, they found different systems, like the exosystem in some insects, grasshoppers, for example. Females are XX, 2 X chromosomes.
Yeah.
But males, just 1 X, they're XO.
And the ratio of males to females stays balanced.
Yeah, because the sperm determinant, half carrying X, half carrying nothing, where the X would be.
Okay.
Then there's the system we're more familiar with, XY. Humans, Drosophila fruit flies.
Females XX, males XY.
Correct. And all the other chromosomes, the non-sex ones, those are called autosomes.
Okay.
And an interesting detail about the human XY system, the Y is much smaller than the X, right?
Yeah.
But they still pair up during meiosis. They have these little matching bits at the ends.
Pseudo-autosomal regions.
Exactly. Pseudo-autosomal regions allows them to pair and segregate properly. And, even before fertilization, there's a subtle thing happening with sex ratios.
Oh, what's that?
Well, it seems Y-bearing sperm might be slightly faster or more successful swimmers on average, statistically.
Really?
Yeah, leads to a slight bias towards males at conception, maybe around 1.3 males for every female conceived.
Huh. I didn't know that.
But that ratio tends to level out closer to 1.1 by the time people reach reproductive age, due to various factors during development.
Fascinating. Okay, so sex chromosomes pointed the way. But for the real, slam dunk proof that a specific gene lives on a specific chromosome, that takes us to the fruit fly lab, doesn't it?
It absolutely does. Thomas Hunt Morgan, starting around 1909, he picked Drosophila melanogaster, the fruit fly, and it was a brilliant choice.
Why them specifically?
Well, they're small, easy to breed, have tons of offspring, really fast generation time, and crucially, they only have 4 pairs of chromosomes. Simple system.
Makes things easier to track.
Yeah.
And then came the discovery.
Right. Morgan found this one male fly that was different. Instead of the normal red eyes, the wild type, this one had white eyes, a mutation.
Okay, so he crossed it.
He did. He crossed the white-eyed male with a normal red-eyed female. The first generation offspring, the F1, they all had red eyes.
Showing white was recessive. Standard Mendel so far.
Exactly. But then he intercrossed those F1 flies, brother-sister mating, and the next generation, the F2. That's where things got weird.
How so?
All the F2 daughters had red eyes, but the sons, half had red eyes, and the other half had white eyes.
Yeah.
It wasn't the expected 3.1 ratio, and it was split by sex.
That pattern just screams sex-linked, doesn't it?
It really does, because males only get 1 X chromosome, right?
Yeah.
From their mother.
Yeah.
The Y chromosome doesn't carry most of the same genes.
So for eye color, there's no corresponding gene on the Y.
Correct. So the male is effectively hemizygous for genes on the X. Whatever allele is on that single X chromosome he inherits, that's the one he expresses.
Whether it's dominant or recessive doesn't matter in the same way?
Right. And this inheritance pattern, where the traits seem to pass from the mother to half her sons, it perfectly matched what you'd expect if the eye color gene was physically on the X chromosome.
So Morgan showed this strong link. But it was his student, Calvin Bridges, who really nailed it down, right? By looking at the mistakes.
Yes. Bridges looked at the exceptions, the flies that didn't fit this neat X-linked pattern. He found extremely rare cases of white-eyed females and red-eyed males in these crosses, which shouldn't happen according to the basic model.
And how did he explain those oddballs?
His genius was connecting it to errors in meiosis, specifically nondisjunction.
Which is when chromosomes fail to separate properly.
Exactly. During egg formation in the mother fly, sometimes the two X chromosomes might fail to separate, so she could produce an egg with two X chromosomes or an egg with no X chromosome at all.
Ah, I see where this is going.
Right. So if one of those abnormal eggs gets fertilized, say a double X egg carrying the white allele on both, gets fertilized by a normal Y-bearing sperm.
You'd get an XXY fly. Who would be female, and if both X's had the white allele, white-eyed? The exception.
Precisely. And Bridges didn't just theorize. He looked at the chromosomes of these exceptional flies under the microscope. He saw the XXY karyotype in the white-eyed females and the rare red-eyed males. They often came from an egg with no X chromosome fertilized by an X-bearing sperm carrying the red allele, so they were XO.
And he confirmed that cytologically, too, seeing the XO chromosome set.
He did. This ability to explain the genetic anomalies by directly observing corresponding chromosomal abnormalities, that was the definitive proof. It cemented the chromosome theory of heredity.
And it also showed something important about Drosophila sex determination, right? That XO fly was male.
Yes. Unlike humans, in flies, XO is male, though sterile. It showed the Y chromosome in Drosophila is necessary for male fertility, but not for determining maleness itself. That depends on something else we'll get to.
Okay, so that's the cornerstone moment. Now we can finally connect Mendel's abstract laws directly to what chromosomes are doing during meiosis. Let's take his first law, the principle of segregation.
Segregation is pretty straightforward once you see it physically. If an individual is heterozygous, say AA, they have one chromosome carrying the A allele and its homologous partner carrying the A allele. During Anaphase I of meiosis, these homologous chromosomes physically separate and move to opposite poles of the cell. That physical separation is this segregation of the A and alleles into different future gametes.
Simple and elegant. What about the principle of independent assortment?
This one depends on what happens just before that. In Metaphase I, imagine you have two different gene pairs on two different pairs of chromosomes. Let's say AA on one pair and blob on another non-homologous pair.
Okay.
When these pairs line up at the metaphase plate, the center of the cell, how the A pair orients relative to the B pair is completely random.
Like flipping two separate coins.
Exactly. The A chromosome could end up on the same side as the B chromosome, or it could end up on the same side as the B chromosome. It's a 50-50 chance for each pair independently.
And because of that random lineup,
when they separate in Anaphase I, you end up producing all four possible combinations of alleles in the gametes, AB, A, B, AB, in roughly equal proportions, about 14 each. That random alignment is the physical basis of independent assortment.
makes perfect sense. But there's a catch, right?
A very important one. This only applies to genes located on different pairs of chromosomes. If genes are on the same chromosome, especially if they're close together, they tend to be inherited together, violating independent assortment. That's linkage, a topic for another time.
Got it. Okay, let's swing back to humans now. We know about X-linked inheritance. How do we see this playing out with human traits, especially diseases?
Well, X-linked recessive traits are much more noticeable, you could say, than autosomal recessive ones.
Because males are hemizygous.
Exactly. A male only needs one copy of the recessive allele on his single X to show the trait. A female needs two copies, one on each X, which is much less likely.
Yeah.
So you see a big difference in how many males versus females are affected.
Hemophilia is the classic example, isn't it? The blood clotting disorder.
It is. The most common form is X-linked recessive. Affected males almost always inherit it from their mothers, who are heterozygous carriers. They have one normal allele and one affected allele, so they usually don't show symptoms themselves.
And this ran through European royalty.
Famously so. Queen Victoria was a carrier, and it spread through her descendants to the royal families of Russia, Spain, Germany. Tsarina Alexandra of Russia was a carrier, leading to her son Alexei's illness, which had huge historical consequences.
A powerful example, and thankfully, the prognosis is much better today.
Oh, definitely. Treatments have improved dramatically, but it highlights how a single gene on the X chromosome can have such impact.
What's a more common X-linked trait?
Red-green color blindness is a great example.
Mm.
It also involves genes on the X chromosome, specifically genes for the photoreceptor proteins that detect red and green light.
And the prevalence difference is stark there, too.
Very much so. Depending on the population, maybe 5 to 10% of males have some form of red-green color blindness, whereas it's less than 1% in females. Again, because males only need that one X chromosome to carry the allele.
Okay, we focused on the X. What about the Y chromosome? Are there Y-linked traits?
There are Y-linked genes, but far fewer functional ones compared to the X. Maybe fewer than 100 on the Y versus over 1,000 on the X.
And what do those Y genes do?
A lot of them are involved in male fertility and development, which actually limits how mutations get passed on. If a mutation causes infertility, it won't be transmitted. And then there are those pseudoautosomal genes we mentioned earlier.
Right, the ones on the tips of both X and Y.
Yeah, because they exist in 2 copies, even in males, one on X, one on Y, they actually follow inheritance patterns that look more like autosomal genes.
Interesting complexity. Okay, let's shift gears slightly. How is sex determined in the first place? In humans, specifically.
For humans and other placental mammals, it really boils down to the dominant effect of the Y chromosome. The presence of a Y chromosome generally leads to male development.
And the evidence for that?
Comes from individuals with atypical chromosome combinations. People with Turner syndrome, who are XO, just one X, develop as females. People with Klinefelter syndrome, who are XXY, develop as males, despite having 2 X chromosomes.
So the Y overrides the X's, in a way. What on the Y is responsible?
It's a specific gene called SRY, which stands for sex-determining region Y. It's located on the short arm of the Y.
The master switch.
Pretty much. The SRY gene produces a protein called testis-determining factor, or TDF. Early in embryonic development, TDF essentially tells the primitive gonads, okay, you're going to become testes.
And when testes form?
They start producing testosterone, the male sex hormone. And testosterone then directs the development of all the other male characteristics.
So what happens if something goes wrong in that pathway?
Well, a classic example is androgen insensitivity syndrome. These individuals are genetically XY. They have the SRY gene. They make TDF. Their gonads become testes, and the testes make testosterone.
What?
But their body's cells can't respond to the testosterone. There's usually a mutation in the gene for the testosterone receptor, which happens to be located on the X chromosome. So despite being genetically male, XY, they develop externally as females.
Wow, that really shows the power of that single SRY gene trigger, but also the complexity of the whole pathway. But this Y-dominant system isn't universal, is it? We saw Drosophila was different.
Not at all universal. In Drosophila, as we hinted, the Y is just for fertility. Sex is determined by the XA ratio.
The ratio of X chromosomes to sets of autosomes.
Exactly. You compare the number of X chromosomes to the number of haploid sets of autosomes. If that ratio is 1.0 or higher, like X female flies have 2 X's and 2 sets of autosomes, 22 is 1.0, it develops as female. If the ratio is 0.5 or lower, like XY and males have 1 X and 2 sets of autosomes, 12, 0.5, it develops as male. Intermediate ratios can produce intersex individuals.
So completely different logic. Are there other systems too?
Oh yes. Birds, for instance, use a ZW system. Males are the homogametic sex, ZZ, and females are heterogametic, ZW, the opposite of humans.
Okay.
And then you have systems like in honeybees called haplodiploidy. Fertilized eggs develop into diploid females, queens or workers, while unfertilized eggs develop into haploid males, drones. No sex chromosomes involved there at all.
It's amazing how diverse the solutions are. But this brings up a fundamental issue, doesn't it? In systems like XY or XO, females have 2 X chromosomes, males only have 1. Doesn't that mean females have double the dose of all the genes on the X?
That's exactly the problem. If females really produce twice the amount of protein from every X-linked gene compared to males, it would likely be lethal. Development is very sensitive to gene dosage, so organisms needed a way to balance it out.
Dosage compensation.
Dosage compensation, exactly. And again, different organisms evolve different solutions.
What do flies do?
Drosophila use hyperactivation. The male's single X chromosome essentially works overtime. It doubles its rate of gene transcription to match the output of the female's 2 X chromosomes.
Okay, so males level up. What about mammals?
Mammals do the opposite. Instead of the male leveling up, the female levels down. This is X inactivation, a concept developed largely by Mary Lyon.
How does that work?
Very early in female embryonic development, in each individual somatic cell, one of the 2 X chromosomes is randomly chosen and basically shut down. It gets highly condensed and becomes transcriptionally inactive for the most part.
Randomly chosen in each cell.
Yes, and once the choice is made in a cell, all of its descendants inherit that same pattern of inactivation. Either the maternal X is off or the paternal X is off.
Which means adult females are mosaics.
Genetic mosaics, precisely. They're made up of patches of cells where the maternal X is active and other patches where the paternal X is active.
Is there a visible example of this?
The tortoiseshell or calico cat is the classic textbook example. The gene for orange versus black fur color is on the X chromosome in cats. A female cat heterozygous for this gene will have random patches of black fur where the X carrying orange is inactivated, and patches of orange fur where the X carrying black is inactivated.
That makes sense. And can you see this inactivated X?
You can. Cytologically, the inactivated X chromosome condenses into a small, dense structure that stains darkly, often seen attached to the inside edge of the nuclear membrane. It's called a bar body. Finding bar bodies was actually an early way to determine genetic sex.
So X inactivation balances the dosage by silencing one X in females. Okay, let's try to wrap this all up. Quick recap.
That's good.
We started with Mendel's abstract factors and saw how work on chromosomes, especially sex chromosomes, and Morgan's Drosophila experiments with that white eye gene provided the physical basis. The chromosome theory was born.
Right, and we saw how the actual movements of chromosomes during meiosis, specifically homologous chromosomes, separating in Anaphase I and non-homologous pairs aligning randomly in metaphase, directly explain Mendel's laws of segregation and independent assortment.
We looked at X-linked inheritance in humans, like hemophilia and colorblindness, driven by that hemizygous state in males. And then we saw the diversity of sex determination mechanisms.
From the SRY gene trigger in humans to the XA ratio in flies, and others like ZW or haplodiploidy.
And finally, the crucial need for dosage compensation to balance X-linked gene expression between sexes using strategies like hyperactivation in flies or X inactivation and bar bodies in mammals.
It really underscores how evolution has found multiple ways to solve these fundamental genetic challenges. The link between a single gene like SRY and a whole developmental cascade like maleness in humans is pretty profound.
It really is. Which brings us to our final provocative thought for you, the listener, to ponder. We've seen that in humans, the SRY gene on the Y chromosome acts as that key trigger for male development. So what do you think might happen genetically, developmentally, if that SRY gene somehow, maybe through a rare mutation or translocation, ended up on a different chromosome, say, an autosome? What would the consequences be?
Something interesting to consider. Thank you for joining us for this deep dive into the chromosomal basis of heredity.
We'll catch you next time.